Everybody should now be able to see my screen. Let's see. I am going to shut off the camera so you don't see the top of my head. And we'll go ahead and get started. So this first slide just has the learning objectives. These are also, remember, on Canvas. So the learning objectives for each one of the chapters is in Canvas itself. It's under the little chapter heading. There's also a summary document of them that is sitting in the exam module. So you've got to access these in a couple places. I always put these at the start of the slide because remember, these are what I use to guide my exam. These are what I think are important, and these are the things that I want you guys to be able to kind of know and do as you're going through the chapter and the information. One thing you'll notice on these ones is that there is this section for previous knowledge. The part of chapter 5 that kind of talks about organelles and what the functions of organelles are, I expect you guys to have had that in other classes. I do expect you to know that. They're fair game for questions on exams, but more importantly, they're going to come into play when we talk about things like bacterial metabolism or when we talk about the immune system. And so the previous knowledge outcomes, you'll notice I didn't talk a lot about in the videos. It's because I expect you guys to know those from past classes. If you haven't found it, there is a link on Canvas in the materials for a biology book. So instead of micro, there's a general biology free access book there. If some of these things that I say are previous knowledge, you're like, that doesn't really seem like previous knowledge to me, go back and check that biology book out so that you can kind of refresh your memory there. For chapter 11, that's really where we're focusing for this chapter, which is really the differences between the various microbes in the eukaryotic category. So as opposed to kind of details of every single microbial eukaryote, I really more want you to focus on what are the big differences between the groups and kind of how do those differences allow you to maybe identify what these things are. So we'll go ahead and move through some of these slides. So this first question is really just going over basic difference between eukaryotes and prokaryotes. And so this is something that you should be able to look at and do relatively simply without too much difficulty. And so if you can't, remember to go back and review those differences. So we want to look at some of these things and say, okay, which ones are found just in prokaryotes, which ones are found just in eukaryotes, and which ones are found in both. You guys should pretty quickly be able to see both DNA and cell membranes found in all cells. Okay, so all cells have DNA genomes, all cells have a cell membrane to keep the insides in and the outsides out. Remember, that's one of the big differences with viruses and why we call viruses acellular is because not all viruses have to have a DNA genome and not all viruses have a lipid layer. And so most of them don't actually have a cell membrane. Okay, so let's see if there's anything else on this list that we would also say was in both of them. You guys should pick out that technically flagella are in both prokaryotes and eukaryotes. This is one of those specialized structures. Not all prokaryotes have a flagella, not all eukaryotes have a flagella. And the flagella that they have is technically different. Since this just says flagella, that structure is in both. But if this had said flagella that moves in a whip-like motion, then that's not both anymore. Okay, and you should be able to stop and think to yourself, okay, which one was it? Okay, those whip-like motions were the eukaryotes. And so while they are there in both, they function for movement in both, the structure of them is a little bit different. So depending on how that's written, it could be both or it could be one or the other if more detail was given. All right, so now let's go through and say, which one of these fit into our prokaryotic categories? If we think back to last week when we did chapter 5, peptidoglycan is part of the cell wall in most bacteria, and so that's in the prokaryotic group. Endospores were one of those specialized structures. And then we have 2 ribosomes here. Remember, there is a difference between the prokaryotic and the eukaryotic ribosome. So we've got to say, okay, which one was it? It's the 70S ribosome that are in your prokaryotes. And that leaves us with the 80S ribosome found in eukaryotes. The Golgi apparatus is one of those membrane-bound organelles. all membrane-bound organelles are really only going to be in your eukaryotes. And then cilia were one of those specialized structures that we found in eukaryotic cells, at least some of them. We don't find the cilia in the prokaryotes. And so that's just kind of a basic overview of different types of properties that can help tell you, are you looking at a prokaryotic cell or a eukaryotic cell, besides the standard nucleus. Okay, so everyone says eukaryotes have a nucleus and prokaryotes don't. There's a lot of other things that are different between those classifications of cells. Okay, moving now more into the actual eukaryotic microbes, so the classifications and the types of microbes that we consider to be eukaryotes. What we have here is various descriptions of different types of eukaryotic microbes. And what we're looking to see is, can we match the descriptions to the actual pictures that we can see here? The key is, I won't ever give you something that just has this name. You guys don't need to memorize genus species names and what they are. I consider that to be what I call Googleable. What we care about in these is the descriptions. Okay? That this is saying that this organism is a dinoflagellate. You should have a general understanding of what a dinoflagellate is at this point. And if you remember, dinoflagellates, so one key part of that word is flagellate for flagella. Okay, the dinoflagellates are the organisms that have two flagella, one going this way and the other one crossing and going perpendicular to it. And so the dinoflagellate that causes red tide is this guy here that's seen in F. It's got the two flagella. As we work our way down, again, we've got here an algal, so an algae. The key property of algae is that they can do photosynthesis. And if we look at these, we have this guy here that's green in color, means that that has chlorophyll, and so that's our algae. Okay. This one is a mold. Okay. Remember, the molds form hyphae. We tend to be pretty familiar with molds because you see them a lot growing on bread and various things that have gone bad. And when they do, you get this nice fuzzy structure. Okay. Those fuzzy structures are the hyphae that are going to form spores and actually blow out into the atmosphere. Okay. So that's our mold there in A. Entamoeba histolytica, we see here is an amoebal disease. Okay. Your amoeba are big. Okay. These are big eukaryotic cells, and their defining feature is that they move by pseudopods. So they kind of stick part of their cell out, and then they can pull it back, and they can use those pseudopods both for movement and to consume food, okay, because these guys are actually going to be predators. They're hunting and consuming things. And so we can see here all of the pseudopods, okay, in this structure, and this little guy, which if you notice is green in color, so it's probably either a small algae or a cyanobacteria, but it's going to be engulfed. So we can see those pseudopods coming out. Hey, this algae is actually going to take this in to consume it and eat it. So our amoeba here is D. Cryptococcus neoformans tells us it's a yeast. One of the key properties of yeast are they're single cellular, so they look a lot like just big bacteria under the microscope. But they're cells that reproduce by budding, which means when they divide, they don't divide in half, but instead they have this little cell that kind of starts pushing out of the big cells, and we can see that budding going there in C. Okay, so that tells us that's our yeast. And then our last one is our tapeworm. Okay, obviously, process of elimination, that leaves us E. But you should also be able to tell, looking at this, tapeworms are large organisms. They're actually not microscopic. And this would be the head. So that's the solix that would actually be embedded in the intestinal wall. And you'll notice you can see the divisions along there. You can see some up here. Remember, those are the protoglottids that actually will break off and release and have the eggs in them that will move into the environment. So as we're looking at these slides, the purpose of this was to get you guys thinking about what are the different types of microbes and kind of what are their key properties that you would use if you have to figure out what one is compared to what another one is. We can do that same thing in written form, okay, so in problems. And these are the types of things that we look at, say, in multiple choice question. So right now I have them written just as true, false, because you can fit more of them on a slide. But really all multiple choice questions is a whole bunch of true falses. Okay, you're just looking at the answers to say, is that a true statement that answers the question or not? So let's look through these, and then I'm going to pause and see if you guys have any questions about these eukaryotic microbes. As we go through the true false ones, I'm actually going to ask you guys to answer them. So I want you thinking a little bit now. So in the chat, I'll just ask you to type in T's or F's depending on what you think the answers to them are. Okay, so if we look at this first one, hey, it's telling us that we've got a drug called a trizol, and that drug prevents the biosynthesis of ergosterol. So the drug is likely an antimalarial that's used to treat the malarial apicocomplexin, Plasmodium falciparum. Okay, so again, remember, I never expect you guys to memorize what trizols do. So this statement wouldn't just say trizols are an antimalarial. Okay? It's going to give you the key information. So it tells us that it's preventing the biosynthesis of ergosterol. Okay? And then we think it's an antimalarial because it would treat apicocomplexin parasites like Plasmodium falciparum. So what do you guys think? Do you think that's a true statement or a false statement? Go ahead and type the answers into the chat. And if you're not sure, you get a 50-50 chance. Okay, so I'm seeing a couple trues, got a false in there. So let's talk about this one. Whoops, sorry, changed the slides on you. What happens when I try to use my mouse on a different screen and I'm on the wrong screen? Let's go back to where we came from. Okay, so this is actually a false statement. Okay, so a lot of people had true up on the chat. So let's talk about why this is false. And these are the kind of the logic you want to think about when you go through these questions. So first, we've got this thing called ergosterol. And so the question you have to ask yourself is, well, what is ergosterol and what type of organism was it found in? Ergosterol is similar to cholesterol. And so animal cells all have cholesterol in their cell membranes. It helps keep the membranes fluid. Okay. In the fungi, remember, they're similar to animals, but they, instead of having cholesterol, they've got another sterol called ergosterol. And so ergosterol, if we're inhibiting the biosynthesis of ergosterol, what we're targeting are the group of fungi. And so then we need to come over and say, okay, we've got here an apicocomplexin parasite. Are apicocomplexins fungi? And the answer is no, they're not. So apicocomplexins were their own type of cell. They were one of the subclassifications of alveolates. And so the drug is not an antimalarial. The drug is actually an antifungal. If we're preventing the synthesis of this fungal molecule, then we're trying to target fungal cells with the drug itself. And so that's kind of the logic you want to look at as you think about what properties might be associated with one cell type over another. Okay, the next question has two major groups of alveolates, are ciliates and dinoflagellates. One of the key differences between these two groups are the structures that they use for movement of the cell. So think a minute about what ciliates have and what dinoflagellates have, and go ahead and type in the chat if you think that's a true or a false statement. Good. So this one is true. Okay, the big difference between ciliates and dinoflagellates is right in the name. Ciliates use cilia for movement and also to kind of brush food into their mouths. Dinoflagellates, remember, have flagellate in there, so they use flagella. They are both alveolates, and if you remember an alveolate, if we look at the cell structure of an alveolate, the main defining feature is this would be the cell membrane, and underneath that cell membrane are these flattened stacks. We call them alveola, or we call them vacuoles. They're just these vacuole membranes that help support the membrane itself. So this is the cell membrane. And these are all just piled underneath it, kind of helping support the membrane and pushing up on it. Those are the key features of all alveolates. So ciliates and dinoflagellates both have this structure under their membranes, but they differ because one has cilia and one has flagella. All right, the third point here, we've got microscopic eukaryotes, such as amoeba and ciliates, are predators that attack and consume other microscopic organisms. I already mentioned that one on the last slide, okay, so we'll say, yes, that's a true statement. Your amoeba, your ciliates are actually eating, they are consuming other microbes, be they microbial eukaryotes or prokaryotes, so they're definitely considered predators in the food chain. And then the last statement is both true algae and second algae must rely on photosynthesis to survive. So go ahead and type in the chat. Do you guys think that's a true statement or a false statement? Good, I'm seeing a bunch of falses show up. Hey, this is a false statement, and that's the big difference between a true algae and a secondary algae. True algae have chloroplasts, and that's all they can use. So your true algae do have to rely on photosynthesis in order to survive and get their energy. But secondary algae became algae because they actually consumed and formed an endosymbiotic relationship with a true algae, a billion years ago. This means that they have the properties of the true algae. They can do photosynthesis, but they were a predator cell before that. And so they also have the ability to pull in other cells as a predator, break them down and consume them for food, kind of similar to you do when you eat food, you're breaking it down and getting energy out of it. So your secondary algae can do photosynthesis, but they can also get their energy from food they consume. So they have a lot more diversity and they don't have to rely on sunlight or photosynthesis to survive. So good, that one is false. Okay, so I am actually going to turn my camera back on for a minute. So does anyone have any questions? We're going to pause right here before we move into the viral chapter. Does anyone have questions about the eukaryotic microbes? I didn't go into every single microbe in this section because you have the video lectures, but hopefully this gave you a sense of how these questions might be asked and what are the types of things that you kind of need to know and think about for each one. I'm more interested, can you tell the difference between the groups than knowing every single detail about them? So questions right now from anyone, you can either type them in the chat or you can go ahead and unmute yourself and just ask. Going once, going twice.
Oh, I have a quick question.
Yep.
So do gram stains, are they only for prokaryotes or can we use them on eukaryotes as well?
Good, that's a good question. So the gram stain are only for prokaryotes because if you remember what a gram stain is really doing is the stain is going to bind to the cell. And then when you do the ethanol step, that ethanol, the reason that the stain stays on your gram positive is because it compresses that peptidoglycan layer. So the peptidoglycan dehydrates and it traps all the purple dye in there. The key is eukaryotes don't have peptidoglycan. So if you did a gram stain on a eukaryotic cell, they would probably change color. So the stain itself could stick to them, but it doesn't actually tell you anything about whether they are gram positive or gram negative because those terms don't exist for eukaryotes. And if you did run the stain, they would all be red. So when you added ethanol to them, you'd wash all that dye away. You'd put your counter stain on, everything would turn red. And all it would do is really kind of just pick up a little bit of stain on the cell itself just because of the charge attraction. So you could do it, but it wouldn't tell you anything because what you're really looking at for the gram stain is what is your peptidoglycan structure.
Oh, thank you.
Yep. Oh, okay. It looks like Chloe answered her own question. She had asked about where ergosterol is. It'll be in the fungal chapters. Okay, anyone else? All right, I'm going to shut my video off again, and then we'll go ahead and move into the virus sections. Okay, so again, learning objectives, these are just there so that you guys can go through them later and use them for your studying. The biggest things to notice as you're looking through these, a lot of the objectives for chapter 12 are understanding what a virus is and what are the different properties that you can have in a virus. So kind of what are the different shapes, what are the type of genomes, and then looking at how viruses replicate. So what are they doing in replication? What are the steps of replication? And how are the replication steps different for the different classifications of virus? So how is a naked virus different than an enveloped virus when replicating? How is an RNA virus different than a DNA virus when replicating? And so that's kind of connecting the viral structures to the replication. So let's go ahead and look at some questions. Okay, so this is just a basic multiple choice question to get us started. Just wants to know which of the following traits is not describing the virus. So we've got that most virions, and I want to point out, because I've had a couple people ask, virus and virion are the same thing. So there are just kind of two different words that describe the same exact viral particle, which is another way we say them. So if I use virion or virus interchangeably, it's because we're talking about the viral structure. So most virions are able to pass through a 0.45 micrometer filter. Different types of viruses can have very diverse metabolisms. Viral genomes can be DNA or RNA, or different types of viruses can contain a lipid bilayer similar to a cell membrane. So go ahead in the chat, A, B, C, or D. What do you guys think is the right answer? Good. So I'm seeing lots of B's show up. Okay. And that's the big key. Remember, viruses don't have any metabolism. So this is really why we consider them acellular, so not actual cells. It's because they have some structure, but they don't have any metabolism. They don't have any chemical reactions happening inside that virion. And so if they're sitting on your desk, they can't replicate and grow on their own. Remember, what a viral structure is going to look like is some sort of nucleic acid. And C is a correct statement. This nucleic acid could be DNA or it could be RNA. And then that nucleic acid is surrounded by a protein coat that we call a capsid. And realistically, that's all you need to have a virus. You need a genome made up of nucleic acid, and you need a protein coat that's protecting it. Really what viruses are is genome delivery systems. This protein capsid is going to protect the nucleic acid until it can be delivered to the cytoplasm of a cell. Some viruses also have a lipid layer along the outside of them. And so this is a lipid bilayer. We call that the envelope. But remember, the envelope is only found in some viruses, and so that's more of a specialized structure. All of your viruses have to have a genome and a capsid. Not all of them have to have an envelope. Okay, here's another multiple choice question we'll take a look at. So bacteriophage, remember bacteriophage will often also just be called phage, so you might hear me abbreviate them as phage. All bacteriophage are viruses that infect bacterial cells as opposed to animal cells or plant cells. Okay, so keep in mind, every species on the planet has viruses that are unique to it. So there are animal viruses, there are plant viruses, there are fungi viruses, there are algae viruses, and there are bacteria viruses. And we just give them a special name because we like to think bacteria are special in our bacterial world. So we call them bacteriophages. So this statement says that bacteriophage have a very narrow host range, which is controlled by the phage tail fiber structures. What's the most likely explanation for this property? Okay, so A is that bacteriophage tail fibers have identical structures, which means that they are able to bind to similar host receptors found in many species. B is that those tail fibers physically inject the phage DNA genome into the bacterial cytoplasm, and that certain tail fibers can only inject into specific species of bacteria. C is that the tail fibers from different phage are only able to bind to receptors found in a limited number of bacterial species. which causes them to infect a small section of bacteria. And then the last one was that the tail fibers actually enter the bacterial cell directly, and the entry can only occur in a specific species, which makes them have a narrow host range. And so as you went through and tried to answer this question, there's a couple of things you need to think about. The first is, well, what are the tail fiber structures and what are they doing? Remember, bacteriophage look like these little lunar module landing things. They've got a head with the nucleic acid, then they have this stalk and these little legs that stick out the bottom. These are the tail fibers. So these are what we're talking about in this problem here. And a host range, remember, is what types of organisms or what types of species are infected by the virus. And so as you read through, the answer to this one was going to be C. Those tail fibers are only able to bind to receptors that are found on a limited number of bacterial species, which means they can only affect a small selection of bacteria, which is a narrow host range. A narrow host ranges mean the virus can only infect one or two species, as opposed to a broad host range, which would say be able to infect lots of different species. The key step for host range is attachment of the viruses. And so that's why we care about the tail fibers here, because those are the ones that if we have a bacterial cell, it's the tail fibers that are binding to receptors on the bacterial surface and doing the attachment. Okay, so remember that attachment step was key for viruses, both for beginning replication, but also for defining what they can replicate in. So what is their host range? What species can they affect? Okay, I'm going to pause there for a minute. Does anyone have questions right now just about kind of the general viral structure? Okay, looks like everyone's all right. Okay, so really no matter the type of virus, whether it's an animal virus or a bacteriophage, whether it's a DNA virus or an RNA virus, the steps of viral replication, so the viral life cycle, are the same. What happens in those steps might be a little bit different, but the general steps are going to be the same. And so we have 5 steps to the life cycle. And this question wants us to go ahead and put those 5 in order. So let's take a look. If we have, here's our cell. Okay, so we'll say this is an animal virus. So this is an animal cell. Okay. The very first step for any virus is going to be attachment. Okay. And remember, we just talked about that attachment is key. It's like a key going into a lock. So there's going to be a receptor on the surface of the cell that has some certain shape to it. And when you look at the viral particle, it's going to have a protein on its surface that fits into that receptor, and that shape is going to be able to bind and interact. So this is that step 1 where we physically have to attach to the cell. Okay. If you change the shape of that receptor, or if the virus changed the shape of that spike protein, that interaction couldn't happen anymore. Okay? And that's going to cause problems. The virus wouldn't be able to replicate. The other way, so the other direction, is sometimes the receptor and the virus can interact, but not real well. So the virus can get in every once in a while, but it doesn't do a great job at it. But then the virus can replicate and mutate and change its spike protein to better fit into the receptor. And then we get better infection. That's actually typically what we see with novel viruses, right? Things like coronavirus right now that has jumped from animals to humans. When that jump happens, the virus is used to binding to the animal receptor. It might not bind as well to the human version of that receptor. But if the virus mutates, so now it recognizes the human better, it actually starts spreading human to human. And so that's what, when they talk at the very start of outbreaks about is there human to human spread, what that means is, has the virus adapted and mutated to recognize our receptors better so it can mutate and get in, and then it can replicate really high and spread from people to people. So this interaction here is really key for viral replication and for what types of species, okay, that host range that the virus can actually infect. Okay, step 2 is going to be the entry and the uncoding. And so this is where that viral particle is going to have to enter the cell. And that can be done in one of two ways. We can either kind of invaginate this membrane, okay, and pull the viral particle in this way. So now we end up within a vesicle in the cell. That's called endocytosis. Or if this is a virus with a lipid layer, that virus gets pulled real close. And what happens, let's see if that's going to allow me to do this. Nope, thought that was going to happen. Never mind. We'll put that back. What happens with the lipid layer is this is actually going, not what I want to do. Sorry about that. This is going to disappear, and this lipid layer is going to fuse with this lipid layer, and the nucleic acid would actually get dumped into the cytoplasm. So this is called membrane fusion. And either one of these, so endocytosis or membrane fusion, are ways to enter the cell. And remember, endocytosis has to happen with your naked viruses, but your enveloped can do either one. Okay. Once inside, if we come in through endocytosis, there are a lot of other mechanisms that'll happen to get that nucleic acid into the cytoplasm. That's the uncoating part. So when we talk about uncoating, we mean we're taking the capsid protective layer away and we're dumping the nucleic acid into the cytoplasm. The next step is going to be gene expression. That is where, oops, that should be a 3. Okay. So our next step is where we're going to take these genomes. We're going to make lots of copies of the genome itself. So we're going to replicate the genome. We're also going to make a whole lot of viral proteins, okay, through transcription and translation. Okay, and that's called gene expression. Remember, this is a little bit different between RNA viruses and DNA viruses, and we'll look at that in the next couple slides. Next step is going to be assembly, where these proteins and the nucleic acids have to come together, so we actually build our viruses. And then the last step is exit, okay, and that can again happen in one of two ways. So if this is an enveloped virus, that virion is going to come over here and it's actually going to push against the cell membrane. So what will happen is that membrane starts to kind of push out. Nope, I wanted blue. That membrane pushes out. Okay, the actual viral particle is here, and eventually this is going to squish together. So we end up with our enveloped virus. And that's where the envelope itself comes from. It comes from pushing through the cell membrane, okay, and that process is called budding. The naked viruses, if this was a naked virus, it actually just destroys the cell, and so we burst the cell open and all the viral particles just spill out into the environment. That kills the cell. Budding doesn't necessarily kill the cell, and so the cell is still alive when the viruses are budding out of it, but now it's just this little viral factory that's not doing its cell jobs, it's just making new viruses. And naked viruses actually kill the cell because that cell has to burst open to release them. Okay, so that's really the overview of all those steps of viral replication. Going to pause there for a second. We're going to get into the differences between the DNA and the RNA viruses. But does anyone have questions about kind of the differences between naked and enveloped and how they get in or how they get out, or kind of these overall steps?
I have a question. Sorry, me again.
Yeah.
So with the naked cell, they already have that membrane, right? The protein, they're already assembled. So why don't they bud as well? Oh,
Okay,
no,
so
never. Yeah.
You answer your own question?
Yes, I did. Never mind. Thank you.
Yeah, that's the big difference. The enveloped viruses have the lipid layer, the naked viruses don't, and so they don't bud because they don't want one of those lipid layers. Anyone else?
I have a question.
Yeah.
So you said that when a virus enters by membrane fusion, is it envelope only or for both envelope and naked?
That's enveloped only. So membrane fusion, because the internal part of lipid bilayers are hydrophobic, they actually, if you bring two membranes close together, they'll automatically fuse to make one. And so if you have an enveloped virus, it has that lipid layer on the outside, which means if you just put it real close to the cell membrane, it'll automatically fuse right into that cell membrane. Naked viruses don't have the lipid envelope, so you could put them as close as you wanted, but there's no lipid layer there to fuse, so they can't use that membrane fusion step.
Okay, thank you.
Yep. Anyone else? Okay, I see a comment over here on the side. Okay, so the question is, when a virus leaves the cell, does it leave to just keep making more viruses, and can the cell still function normally after? The cell can't normally function after. So even for the budding viruses, as they're budding out, what's going to end up happening is the virus has really taken over the ribosomes of the cell. So instead of making cellular proteins, the cell is now making viral proteins. And so what ends up happening is all of the chemical reactions that your cells normally need to do. So for example, if you're a liver cell, you have to be doing things like making glycogen or breaking down glycogen to control blood glucose levels. Those enzymatic processes can't happen if you're not making the enzymes that you need to. And so as the viruses are budding, that viral replication process is still ongoing. And so the cell never goes back to normal. Pretty much what ends up happening is that little viral factory of our cells gets killed by our own immune system to stop the virus production. And so we kind of kill some of our own cells, even if the virus isn't, just to make sure that they're no longer producing those viruses. So whether they're lysed or budding, the cell itself is going to die in the long run. It can't go back to normal. Hey, anyone else? Okay, so there's just two more questions on here. So let's go ahead and work our way through. The next one is one of those true-false sections, so I can fit a couple into one. And these are really just looking at conceptually, do you understand the differences between the different types of kind of terms that we've been using in the viral chapter? Okay, so this first question, we've got a lytic phage infects bacterial cells similarly to how latent viruses infect animal cells. So go ahead in the chat. Do you guys think that's a true statement or a false statement? Okay, good. I'm seeing a bunch of falses come up. And that's a false statement. So first off, a lytic phage, remember there's two types of bacteriophage. We've got lytic cycles and lysogenic cycles. Okay. Lytic phage can do the lytic cycle. What we call temperate phage can do the lysogenic cycle. Okay. Latent viruses, those are the ones that will get into animal cells and go dormant. So they'll actually just kind of let their nucleic acid hang out. It either becomes part of the genome of our cells or it just hangs out in this little circular form, but it's not replicating and making new viruses. So we could actually change this statement to make it true by changing our lytic phage to be temperate phage. So it's the lysogenic cycle for bacteriophage that's similar to latent animal viruses. Okay? They both are able to integrate into the genome, hang out inside the cell for a while without making viruses, and then at some point in the future, kind of pop back out, start making active viruses, and then they cause problems. Okay, so your lysogenic cycles in your bacteriophage are similar to your latent replication cycles in your animal viruses. All right, well, again, we'll move on to the second one. The fact that influenza virus is able to bind to the respiratory epithelium, but not to liver cells, describes its tissue tropism and is controlled by its spike proteins. You guys think that's a true statement or a false statement? Good. Seeing lots of trues show up. And we've already talked about the spike proteins and their role in attachment. Remember, attachment is important for your host range, what species you can infect, but it's also important for the tissue tropism, which is once the virus gets into a body, what types of tissues can it actually interact with? Okay, those receptor proteins on the surface of cells are not found on every single cell type in a body. It's why you're a multicellular organism, and your organs do different things, which means they have different proteins. And so this attachment step with those spike proteins controls tissue tropism and your host range. As next one, a filamentous virus contains a helical-shaped capsid that will extend the length of the viral genome. And again, this is just terminology about kind of descriptions of your viruses. And this one is a true statement. And so you guys should give a general sense of the difference between a filamentous virus and a icosahedral virus. Hey, one thing I will note, I actually had someone ask in the viral concept map, one of the words in your answer key was a complex virus, but it was not a word that was in the lecture notes. So your bacteriophage, those big tailed structures, are sometimes called complex. So just to kind of clear that question up, complex viruses that you might have seen in the concept map are the tailed bacteriophage that we talked about in the lecture notes. Some of that is just different terminology in different textbooks. So I apologize for that confusion. And now the last one here is you've discovered a novel virus and find that when you infect specific cells, the cells are lysed by the virus. Hey, this tells you you're working with a naked virus. This is what we were just talking about. This just shows you one of the ways that I can ask those types of questions. Hey, and so that's a true statement. Hey, if you're seeing that the cell is being lysed, we know we're working with naked viruses. Okay, now the last question. This is the one, I saved it to the end because this tends to be where students end up with questions when we're looking at the viral chapter. So I'm going to have you guys answer this one in the chat. It says, you've been working with a novel virus to identify its physical properties. When you're studying the viral proteins in the virion itself, you found an RNA-dependent RNA polymerase as part of the virion, so packaged as part of the virus. What does this tell you about the virus? A, it's a positive sense RNA virus. B, it must be a double-stranded DNA virus. C, it's either a negative sense RNA or a double-stranded RNA virus. Or D, it's a double-stranded or single-stranded DNA virus. So I want you guys to go ahead and answer this one in the chat. What do you guys think? Good. So I'm seeing lots of C's show up. Now here's the key. If I asked you why, do you guys feel like you would be able to explain that question? Or have you just memorized that RNA-dependent RNA polymerases are part of the virion for these and not your positive sense? Because it's that why and that explanation that's going to be important. And so do you guys want me to go over kind of the why, what the differences are, or does everybody feel comfortable about the why for that? Okay, I'm seeing a couple of yeses to that question show up in the chat. So let's walk through it. Okay, and then see why wouldn't it be a positive single strand? And so we're going to take a look at that. So here's the key when we talk about viruses. And this is one of the things that is a little bit complicated when I skip chapters. So you'll notice we pretty much did chapters 3 and 5 and then jumped all the way to 11 and 12. It's because I like to do the overall survey of all of the microbes at the start of the semester. But because we've skipped to later chapters, the kind of genetics chapter we haven't really covered yet. So this should be background knowledge. This should be things you got in general biology, but not everyone always remembers it. And so the key to understanding the RNA viruses is remembering that when you learned about molecular biology in your general biology class, we've got DNA, right, that's a double-stranded molecule, and those strands go in a certain orientation. So if we looked at the structure of DNA itself, there's a 5' end, which is actually where there's a phosphate on the structure, and there's a 3' end, which is where we get the end of the sugar molecule. Those are anti-parallel, so we go 5' to 3' in the other direction. That DNA gets transcribed by an enzyme called RNA polymerase to make messenger RNA. And here's the key. Messenger RNA goes 5' to 3'. And this then gets translated. And remember, that happens by the ribosomes to make some sort of protein that's going to have some sort of 3D shape. And so the key here is that ribosomes will only recognize messenger RNA that goes from 5' to 3'. This is what we call positive sense. Okay? It's what the cell, all of your cells, all of every cell. So what we have drawn here happens in animals, plants, fungi, bacteria, archaea. This is the central dogma for what we know as cellular functions. We take DNA molecules, we transcribe them into messenger RNA that runs 5' to 3', and we translate that into proteins that can do stuff in the cell. From an RNA virus standpoint, they have to be able to fit into this pathway somehow, or they can't hijack our cells. But here's the key. So positive sense single-stranded RNA viruses, their genome looks like this. And you should notice that those look very similar. Negative sense single-stranded RNA genomes go in the opposite direction. So that's 3' to 5'. And then your double-stranded RNA genomes are similar to your DNA in that they are anti-parallel. But what you want to notice is that neither one of these structures exist over here in the normal central dogma, which means when your negative sense or your double-stranded RNA get into the cell has no idea what to do with them. They have never seen at any point during their cellular life a molecule that looks like this or looks like this. And so in the cytoplasm, they would just get destroyed. They just get degraded. And so what happens is if you are one of these types of RNA viruses, you have to be doing something so the cell doesn't destroy you and so you can actually be replicated. Okay. So here's what it looks like when we get into the cell. Your positive sense RNA viruses, because they look like your messenger RNA, these immediately are going to be translated into protein. Okay, the ribosomes in the cytoplasm see this, think it's messenger RNA, and immediately translate it into proteins. The key is that these are viral proteins, and one of them is that RDRP, okay, that RNA-dependent RNA polymerase, because that RDRP gene is somewhere in the genome here. Okay. We say that's encoded. Okay, so the RDRP is an encoded gene. It's part of the genome. And when this gets translated by the ribosomes, we make that as a functional protein. That RDRP can now come over, interact with the genome itself so that we can do step 2, which is replication. So now we're making lots more of your viral RNA. And remember, that was the gene expression step. Now we've got the viral proteins, the viral RNA, and we can start with the assembly. For our negative sense and our double-stranded, the ribosomes don't recognize these. And so we can't start with step 1 being translation. And because of that, in the virus itself, we have to have that RDRP enzyme, so the protein, already made. So that when this enters the cytoplasm, what's entering the cytoplasm is both the genome and the RDRP enzyme. And so that RDRP is already there to interact with the genome. And now what we do is step 1 is replication. One of these genomes is actually made 5' to 3', so that now that looks like messenger RNA. And we can do step 2, which was translation to make all of our proteins. Okay, so now we again have the RNA genome and the proteins, and we can do the assembly step. Okay. The difference was we had to do them in the opposite order because our cells, so no cell, will translate this negative sense or double-stranded RNA. So instead, we need to replicate it first and switch its orientation. So now the cell will recognize this to make all of the viral proteins that we need. What that means is if you look at the virion itself, so if we looked at the virus, we would have a capsid. We would have, say, that 3' to 5' genome. Okay, so we just had drawn already. We've got the nucleic acid surrounded by the capsid. But as part of the virus itself, we would also have that RDRP enzyme. Okay, so that explains kind of the why behind why these two types have to have RDRP as a fully formed protein, whereas your positive sense RNA viruses, they have the gene for it, and they will make the protein eventually, but it doesn't have to be part of the virus itself. The virus itself for these guys will have the capsid and the 5' to 3' genome, but it doesn't have to be packaged and be part of the virion. Okay, so I'm going to pause there. I'm going to go ahead and turn my camera back on and see if you guys have any questions. So either about the stuff we just talked about or anything from either one of these two chapters to start with. So conceptually, information, material type questions. Okay, so I got a question here about what happens with the double-stranded RNA. The same thing. So I just used the negative sense in the picture, but the same exact thing that I drew for the negative sense will happen with the double-stranded RNA. It's just a little bit more complicated to draw since there's two strands, but conceptually the same thing's happening. The RDRP would replicate it. We would end up with some versions that were 5' to 3', and then those would go ahead and be translated. Any other questions? Going once, going twice. Okay, so I'm going to stop sharing my screen for the moment. And now I'm going to open it up. Do you guys have any questions at all? So exams coming up this weekend. Remember, it's got two parts to it. So one of them is just kind of your standard multiple choice type exam. You've got 2 hours to take it. It's 35 questions. The second part is a little bit more complicated because it's different than a lot of students have done, where you've got this kind of case to work through individually and then answer questions about it. And it's very similar to the case study quiz that you're doing, but those case study quizzes, you can work in a group. Those are more practice for the case study portion of the exam that you have to do individually. So let's see, Chloe, you've got your hand up.
Yeah, so the case study questions, like the case study test, it's not off of the case study we did this week. Is that correct?
Right. So the case study quiz is off of the case study you did with your group this week. And so you'll want to probably take that quiz before you do the exam to make sure that you kind of were on the right line of thinking. The case study for the exam is going to be similar, but it's a different story. And so it's actually walking you through a true story, and you're going to get bits of that story for every one of the exams. But mainly what I'm looking to see is I really want you guys being able to use and apply information in the class. And so the case study assignment and the case study quiz are how to work with your group to learn to do these things. You then get a completely separate scenario for the exam to see individually, have you learned how to use and apply the information? Let's see. Oh, I've got a question in the chat about retroviruses. And so let me actually, I'll go back. Well, let's see if I can answer this one without sharing the screen again. Good. And so the question is, retroviruses, does the material replicate in the nucleus? And non-retroviruses, it replicates in the cytoplasm only. For your RNA viruses, yes. So Retroviruses have an RNA genome also, but they're their own separate category. It's because the 3 RNA genomes we just looked at, the positive sense, the negative sense, and the double-stranded, those are all going to be replicating just in the cytoplasm. They never have to enter the nucleus because they only need to use our ribosomes. The retroviruses, those are the ones that come in as RNA, but they take that RNA and they reverse transcribe it. So they take RNA and they make DNA out of it. That DNA then goes into the nucleus because that's where all the cells house the DNA itself. And so that's true statement. In retroviruses, it enters the nucleus as DNA. In all of your other RNA viruses, it stays in the cytoplasm. Other questions? All right. Going once, going twice. Okay, so remember the exam opens Saturday morning. It actually opens at midnight. Not that I expect anyone to be online at 1 a.m. taking the exam, but it opens Saturday morning. It closes Monday evening, so Monday by 11:59. Just make sure if you're taking it on that Monday, you give yourself enough time so it will kick you out at 11:59. So don't start it at 11:30 thinking you'll get your full 2 hours. Okay, make sure you give yourself time at the end of the day on Monday if that's when you're taking it. Okay, if there are no other questions, I'm going to go ahead and stop the recording, and you guys are free to head on out and enjoy the rest of your afternoon. Remember, I do have office hours this afternoon, so if anyone has individual questions, you can go ahead and sign up for those. See you guys.